In this video, we're going to look at the challenges with experimentation and observational research methods. So what things should we know about experiments and observational studies that should cause us to question the findings? What do we need to make sure we consider as we are constructing research methods that are observational or experimentation? So we need to think about confounding variables. So don't make the mistake of accepting causation, especially with observational studies. Remember, we've talked already about the fact that observational studies do not tell you about causation. In experimental design, we attempt to identify causation, but that's only the case if we can control for all of those other variables. So why observational research cannot tell us about causation is because there may be some other variable, some confounding factor that is causing that is creating a relationship when one is not actually causing another. But there's some other unknown variable that is related to both the explanatory and the response variable. So, for example, in the 19th century, when the Royal Navy's Arctic expedition first visited the North Pole, it was believed that scurvy could be prevented by good hygiene on board the ship, regular exercise, and maintaining crew morale. Well, it turns out that the reason the Royal Navy's Arctic expedition didn't suffer from scurvy was really not related to morale, but it was due to the fact that they ate fresh fish. Later expeditions didn't eat fresh fish, even though they had good hygiene, regular exercise, but they ate instead tainted food. And so the challenge, of course, with observational is because we can't control for everything. We're not choosing who's getting the treatment and who's not. That there are these underlying or confounding variables that could lead us to believe there is a relationship or no relationship when that is not the case. You know, the most, known research in terms of scandalous research is that, and that many people believe today, right, is that immunizations cause autism, right? Well, where did this come from? This came from a poorly designed research project that didn't have mechanisms to control and identify confounding variables. And it has an impact because decades later, people still believe that immunizations cause autism. So when you're designing your research, when you are reading other people's findings, if it is an observational study and they are saying that one thing is causing another, you should immediately know that that is bunk and throw that out. If it is an experimental design and they are looking at something causing something else, you should really question what are the confounding variables? What else might be impacting the response variable? And did they control for that by the way they allocated who's in which sample in order to identify that impact and separate it from the impact of that explanatory variable that they're trying to research? So researchers may try to control the known factors involved with the outcome of interest. That's going to reduce the confounding. So if we're studying the link between blood pressure and work, researchers may measure and take into account factors such as age, diet, smoking history. And so we want to then put people with similar ages in the two groups for diet, smoking, and so on. So confounding is not an issue in experiments if we can control for those particular issues by the way that we allocate individuals to those groups. Another problem with experiments and observational studies is that we may extend the results inappropriately. We've been talking in these videos about how in statistics what we're trying to do is take information from a smaller subset and extrapolate or infer to the greater population. So what we need to do as we look at research is look at who are they saying that this findings are for. So when you choose your sample for your experimental design, for your observational study, your extrapolation to the greater population is going to be limited as to how well you represent that greater population. So our fundamental rule for using data for inference, right, still applies, that if it's not a representative sample of the population, then you're not going to be able to extend your results onto the greater population. With our observational studies, we are using specially selected subgroups. That's going to limit our ability to draw conclusions about them. Make sure that you recognize the sampling frame, the process for selecting these individuals, that sampling procedure. Can you make it as close as possible to the population? And in any of your research reports, make it very clear how you chose your sample, your process for that, and any limitations on that extrapolation to the greater population. When we think about like medical research and experimentation, when we do we test drugs, we test them on healthy volunteers. Well, in the real world, the people who take medications are not the healthy people. And so there is a limit there in extending the results. One of the challenges we have is that we don't do medical studies for drugs and the impact of treatments on pregnant women due to the concern of impact on the fetus. But then that also means we have no information as to when we get to the real world and you want to give a drug to a woman of childbearing age, what the impact will be. So we need to make very clear any assumptions, any limitations, any restrictions on that extrapolation to the greater population. The other problem with experiments and observational studies is interacting variables. So sometimes a second explanatory variable will interact with the principal explanatory variable in its relationships. So when we do, for example, a regression model, so we are trying to predict how high the trees are going to grow. And one variable we have is how much rain they're getting. And another variable is how acidic the soil is. In these models, we often assume that these variables are independent of each other, right? The impact that the acidity of the soil has an impact, and its impact is completely separate from how much water it's getting, how much sun it's getting. Whereas in real life, right, there's likely an interaction between the two. It's the combination and the environment. And so when we design our models, if those explanatory variables are not completely independent of each other, then our model needs to also include that interaction. And trying to figure out how that interaction looks like can be challenging. There is interacting variables in observational studies and in experiments, and they need to be taken into account. So let's just take a look at some examples. All right, in 1994, a study was conducted that recruited 240 smokers at the Mayo Clinic. Volunteers were required to be between ages 20 and 65, have an expired carbon monoxide level of 10 parts per million or greater. So showing that they're a smoker, right? So when they exhale, the level of carbon monoxide is a certain level. At the same time, they need to be in good health. They should have a history of smoking at least 20 cigarettes per day and be motivated to quit. So we're trying to control for a number of factors here to deal with those confounding variables, right? So we're making it so they fall into all these categories. So volunteers are randomly assigned to receive either a nicotine patch or a placebo. So what do we find here? We find that if they're a smoker in the home, then there's less of a difference between the nicotine patch and the placebo patch. And if there is no smoke in the home, then there is a difference. Well, when we did the study, it didn't look at, so it looked at if they were in good health, that they've been smoking, that they were motivated to quit, they were a certain age, and then we gave them the treatment. There's this other variable here, whether there's other smokers in the home, that has an impact. And so we have another variable, but on its own, that variable may not be enough. It's this interaction with these others that causes the effect. So we want to make sure that we don't have confounding variables, that all variables are identified and controlled for or part of the experiment. And then we need to recognize that perhaps variables can have an impact when they are in combinations with each other. Other problems with experiments and observational studies. We talked in previous videos about the Hawthorne effect, right? This is where when people are being observed, it changes their behavior. So the Hawthorne effect, this comes from a study that was done in the 1920s of factory workers at the Western Electric Company in Illinois. They were trying to determine which working environment would make workers more productive, so they were changing the lights. And what they found was that no matter what they changed the lights, the workers got more productive. And it was the fact they were being observed that changed those results. So the concern with the Hawthorne effect and experimentation is that the process, the fact that we are observing them, recording information, would cause them to change behavior. So if we think about it this way, if you are involved in a study and we are asking you to do a workout routine, are you going to do more of that workout routine because you want to look good in front of the researchers than you might otherwise? So it's going to affect the generalizability of the results. Are we going to actually be able to extrapolate this to the greater population? Because the behavior is being changed not because of the treatment, but because of people being observed in the experiment. We also have to be concerned about an experimenter effect. That is when the researchers record the data to match the desired income. So if the researcher treats the subjects differently based on whether they're getting the treatment or the placebo, then that is going to cause issues. There, the experimenter is consciously or unconsciously creating a bias to help kind of push the results that they want. So this is why we have double-blind studies, so that even the researcher themselves doesn't know who is getting the treatment or the placebo, so their behavior doesn't change. So the experimenter effect, the researcher's behavior changes. The Hawthorne effect, the participants' behavior changes, all because they are involved in the research project itself. Another thing we need to look at when it comes to experiments and observational studies is ecological validity and generalizability. So when the variables have been removed from their natural setting and are measured in a lab or an artificial setting, does that change how the results versus if it was in the complicated real world? So there we come back to interactions between variables. In an experimentation, we control for everything, right? The only difference between your two groups, the only difference between your measurements over time is whatever you've chosen as your treatment. You're isolating it so you can see if it causes an impact, which means nothing else is different, nothing else is changing. But in real life, lots of things are changing and they're all changing at the same time. And so this interaction between all these different changes can have unexpected results. And that's why when we do like medical tests in that controlled environment where we have chosen healthy people in a certain age group and a certain behaviors, we've controlled all these different characteristics and we've just given them the drug or not. we then get different results when we go out in the real world and other people are different levels of health. They are different. They're taking other drugs, and suddenly we realize that there is an issue when you take this drug and you take this new drug, and now it creates complications or issues. So we're talking about taking something out into the real complicated real world versus that controlled environment. How valid are the results in that complicated real world? All right. I forget where this list ends. I think this is the last one. All right. Other challenges with experiments and observational studies. When we are doing observational studies in the medical realm, so we're using your medical history, we're looking at that retrospective study. There are issues in using that past as a source of data, a challenge with our observational studies, and it's similar to those challenges that we have with secondary data in general, which is, does the old information represent the new situation? Does the old information collect enough information for what we need? If we are asking people to recall things about the past, there's issues with their recall and the accuracy of that. So problems with recall bias when we're asking people to tell us about things that happened before. So challenges in terms of using data that might have been collected for a different purpose or asking about information that happened in the past. All right, so we've gone through a number of problems with experiments and observational studies, whether you're combining or you're using past data, where you're extrapolating to the greater population, you're concerned about bias from the researcher, changes in behavior by the participant, interacting variables, so complications of things in the real world and other variables that you may not have included in your study or controlled for, and the fact that you didn't control for them at all, so that they are confounding, or we're trying to extrapolate inappropriately. So there are a number of challenges with doing experimentation and observational studies that you need to take into consideration as you're designing your research project.